Hello, everyone. I would like to welcome you all today to the International Journal of Clinical Research. IJCR is an organization rooted with an aim to facilitate and promote high quality research, publication, and interactive clinical content for physicians, educators, and the global medical community. Part of this initiative is the Pioneers in Medicine webinar series. My name is Peter Samuel. I will be your host for today. I'm a fifth year medical student from Georgia. I'm currently a member of the Medical Student Advisory Board and the host for the podcast Medicine Beyond the Science. I now invite Hamza, a third year physical therapy student from Antoine University, Lebanon, a member of the Pioneers in the Medicine Student Chapter. He will be introducing Tijal now.
Thank you, Peter. Hello, everyone. My name is Hamza Deeb. I'm a third-year physical therapy student at the University of Antoine in Lebanon and part of the webinar series student committee. It is my utmost honor to be presenting the International Journal of Clinical Research today. So, what is IJCR? IJCR is registered as a non-profit organization in Delaware, USA, where it has zero article processing charges. However, this has no hindering effect on our high-quality, open access, double-blinded, peer-reviewed articles and interactive clinical content for the global medical community. As for the aims of the journal, establish equity in the publishing field by waiving publication fees and making sure that no authors are financially disadvantaged. Guarantee high-quality publications by employing rigorous editorial, peer, and statistical review processes to evaluate manuscripts. Encourage and promote scientific research by involving young researchers and medical field students like myself, teaching and guiding them through the process of publishing. And finally, to improve patient outcomes by optimizing information delivery, especially from developing countries. Within its scope, various types of research papers are published, including case reports, case series, narrative reviews, and original research. Due to its system and the quality of its articles, IJCR has obtained indexation from ISSN, DOI, Crossref, Google Scholar, Publins, and DOAJ, and it's through the process of obtaining PubMed and Scopus indexations. Although IJCR is so young, starting a couple of years ago in May 2020, when our reach has started from 5 countries and 9 cities, it has achieved exponential growth, and now it reached 131 countries and 1,607 cities. This is the data presented by the Google Scholar Analytics. As for IJCR's organizing committee, we have the managing editor and CEO, Dr. Gregory Nicholas, the assistant managing editors, Dr. Shivanji Saha and Dr. Ricardo Vieira, along with the students' assistants. The editorial board consists of multiple editors from different fields of different specialties. They focus not only on obtaining great quality publications, but also on guiding our authors throughout their publication journey. We have esteemed editors from various countries who highly contribute to the maintained quality of the journal. We are more than proud to say that we have started the board of advisors, and one of them is Professor Barakat, who is the Dean of Undergraduate Studies at the University of Sao Paulo in Brazil, and also happens to be the mentor of Dr. Gregory Nicholas. And here, I would like to mention that we are looking for esteemed faculties to join us as the board of advisors and be a part of this growing family. As mentioned before, in IJCR, diverse kinds of research are published. For instance, as you can see, these are a narrative review, an original research example, and a case report that has been already published. We have 4 divisions at IJCR: the webinar series, the podcast, the course, and the Medical Student Advisory Board, also known as the MSAB, each of which will be discussed in a second. Starting with the webinar series, its main focus is to inspire young minds by showing them the accomplishments and research of great experienced researchers and how they impacted the world at large. This series has been prospering thanks to our director, Ms. Hiba Sati, along with the assistant director, Dr. Keval Thakkar, and our event moderator, Peter Samuel. And I am very proud to be one of the members of the student committee along with my colleagues, Gracia, Abdel Razak, Noof, and Noor. In the webinar series, we have 2 divisions: Pioneers in Medicine and Future Leaders in Medicine. Pioneers in Medicine is a series featuring world's premier experts in their respective fields to discuss their research, medical journey, and inspire the next generation of students to engage in research. Future Leaders in Medicine features early career physicians and researchers with great innovative achievements to discuss their work and share their advice with the upcoming generation of the students. Our first webinar was The Road to a Chemo-Free World with Dr. Bruce Cheeson. Then we had Dr. Alex Kansis presenting Why Do Young People Get Cancer? After that, we were honored to have Dr. Robert Peter Gale, an expert in leukemia and bone marrow disorders. Heading to the fourth webinar with Dr. William O on precision oncology. In December, we had Dr. Patrick Hugh as a keynote speaker on using the immune system to fight cancer. Then we got the chance to enjoy Dr. Razel Gurzurok's speech about genomics and immunotherapy. In addition, we were privileged to hear Dr. Mace Rothenberg, the former Chief Medical Officer of Pfizer, talk about his career in medicine. Most recently, we had the pleasure to host Dr. Naji Riyashi in an amazing webinar about neurology. And today, we are ultimately excited to host Dr. Mary Mitrani, the Chief Science Officer and co-founder of Organicell Regenerative Medicine. We are eagerly waiting for the webinar with Dr. Mary. We also have our podcast, Medicine Beyond Science, hosted by Peter Samuel, a fifth-year medical student at Tbilisi State Medical University in Georgia. We have had 8 podcasts interviewing medical personnel, patients, researchers, and special guests to highlight their unique stories, personal experience, and contributions to medicine. They are on all podcast channels, including Spotify, Apple Podcast, Listen to Notes, and many more. So tune in and make sure to listen to the great stories behind each person and check them out if you haven't yet. Our last and one of our biggest divisions is the Medical Student Advisory Board, which is run by the AMSAB Council, including our advisor, Dr. Nancy Emanuel, and the presidents, Janas Jalbout and Perla Ghalloub. The vice presidents, the executive assistants, the chief administrators, the admission officers, and the research officers, who are responsible for creating, maintaining, and updating a database that includes all the students who are interested and willing to take on positions in research working groups. A research working group is a group of students working on writing a scientific article guided by mentors provided by IJCR. In about 6 months, more than 100 students have been given the opportunity to participate in a research opportunity with 34 ongoing research projects. In the AMSAB, we have more than 100 medical students and fresh graduates from 30 countries. As you may notice, students in this chapter come from very diverse backgrounds. The AMSAB division is divided into the following 9 chapters, which will be explained briefly. First, we have the Journal Club chapter that provides a Journal Club meeting every 2 weeks, which is exclusive to the members of the AMSAB. This chapter teaches students how to write and critically analyze scientific information, while allowing them to develop their presentation skills. Secondly, the Digital Health chapter, responsible for teaching and spreading awareness about digital health, an essential and growing field that each physician must know about. In addition to the webinar series, the student committee helps in organizing the webinars that we have already discussed. The Growth and Development chapter offers every month 1 session of community building activities and a workshop on growth and development. We also have the Social Media chapter, which is responsible for the image of the journal on social media. It teaches students how to manage a professional account and build teamwork and leadership skills. Hands-on writing skills, in collaboration with IJCR Intersect, provides students with a hands-on writing experience, through which students develop their writing skills directly with a mentor. The International Students Outreach chapter gets external collaborators and educational services that IJCR offers for free for associations internationally. Furthermore, the Junior Mentors Program is a research course developed especially for the members of the AMSAB to guide them from beginners to advanced levels. Last but not least, the USMLE Study Group, which is a newly launched chapter, aims to guide, assist, teach, and motivate students who will be taking the USMLE exams to apply for the match in the USA. As you can see, the AMSAB has all these amazing opportunities and much more. If you are interested in joining us, which is highly recommended, follow us on social media and check out the website for further information on how to subscribe through our website, as the applicant subscriptions are now open. Thank you so much for your attention. I'll leave you now with Peter.
Thank you, Hamza, for introducing the journal. As the term international would imply, we have a team of representatives from all around the world. The latest wing of this chapter is the Future Leaders in Medicine. As part of this series, we have a very special speaker joining us today, Dr. Mari Mitrani. I now invite Dr. Keval Thakur, the Assistant Director of the webinar chapter, to introduce the speaker for the evening.
Thank you so much, Peter. In IJCR, we believe in the free and equitable distribution of knowledge. We also firmly believe that the youth is our future, and it is directly intertwined with research innovations. Therefore, we launched a webinar series and initiative where we bring to you medicine's most prominent luminaries discussing their medical journey, research discoveries, and their passion and hope that it will inspire you. Our success cannot be achieved without a great team, and I'm fortunate to have a passionate, hardworking, and dedicated team to make this event a reality. Dr. Mitrani is the Chief Science Officer and co-founder of Organicell Regenerative Medicine. Organicell is the leading fully integrated biologic medicine company where world-class research, technology, manufacturing, and clinical development team focuses on creating new biological medicine. Dr. Mitrani graduated as a medical doctor from Universidad San Francisco de Quito and also completed her PhD in neural therapy from Medical Society of Natural Therapies. She received her Doctor of Acupuncture degree from the Pan American University of Natural Medicine and was appointed as a missionary medical doctor by the Sovereign Medical Order of the Knights Hospitaller. Dr. Mitrani also co-founded the American Cellular and Anti-Aging Center in Quito, Ecuador, one of the first autologous stem cell centers in South America, and along with her husband, opened additional stem cell clinics in Guatemala, Trinidad and Tobago, and Jamaica. She has participated in missionary medical work, and as her primary motivation has always been caring for others. She provided medical attention and also was the translator of medical groups such as Canadian Association of Medical Teams Abroad, Medical Mission Ecuador. For the past 13 years, she has led Organicell in developing innovative treatments to various health conditions. Over the past few years, she has built a team of forward-thinking scientists and researchers and has received several FDA approvals to conduct various trials related to COPD and knee osteoarthritis. I will be briefly highlighting some of her numerous inspiring achievements. Dr. Mitrani has an FDA-approved investigational new drug to her name called Zofin for the treatment of prolonged COVID-19 symptoms. Zofin is an acellular biologic therapeutic derived from perinatal source and is manufactured to retain naturally occurring microRNAs. Not only that, but she has also been recognized as top 100 healthcare leaders in the year 2020-2021 by the International Forum on Advancements in Healthcare for her spearheading work during the COVID-19. In the year 2016, she was felicitated with a humanitarian award to benefit the victims of the Ecuadorian earthquake issued by the Ecuadorian National Congress. Featured on the Wall Street Journal and the Forbes magazine, let us all hear from an inspiring and exceptional role model today. Dr. Mitrani, the floor is yours.
Thank you so much. Thank you for the entire team being here. Good morning, good afternoon, good evening, wherever you are. This is very exciting. I gotta congratulate you and your entire team. You are doing incredible. I am a woman, I am a mother, I am a Latin fierce physician. I'm a physician scientist that understands how important it is representation in medicine and how important it is to see that you are really pushing and giving so many young adults the opportunity of growing into this field, which is an unbelievable field. Anywhere and everywhere that you decide to do in medicine, it's always super, super important. We need you. We need all of us to keep growing together. So I'm gonna share my screen and I'm gonna ask the question that always has to be asked. Can you see my screen once it goes up?
Yes, we can.
Okay, thank you. So let me put on the presentation. And today I'm going to talk a little bit about, I love this subject. This is what motivates me every day, is how cell waste became the next generation of regenerative medicine. As you presented me, I am a medical doctor. I call myself a physician scientist because I am conducting research, right? And as an MD, there's so many physician scientists out there that are in different fields conducting research. So I do also, I'm very passionate about nutrition. I got recently my plant-based nutrition certification, because there is no health when you have not addressed nutrition. And unfortunately, we live in an era where when nutrition is just nobody wants to care about it. And we can cure so many diseases, we can reverse so many diseases just if we pay attention to nutrition. So I do have a PhD in neurotherapy, as you presented. I have a doctor in acupuncture, and last year I was awarded, or it's 2 years, COVID, this top 100 healthcare leader award from IFAH, which was a surprise for me, but very humbling to receive this award. As a physician scientist, I keep up all my certifications every year because I am working on two big things, which is on one side manufacturing biologic drugs, and another hand is conducting human research trials. This is what some of the certifications that I carry every year. And I was recently interviewed by CanvasRebel. One of the questions they asked was very interesting, because they asked, if you could go back, would you choose the same profession and specialty? So I just put a little piece of my interview here. I've left the link in my presentation as well for CanvasRebel interview. And, you know, I did not think I was going to become a doctor until I started getting biology, anatomy, and physiology in my 10th grade. And I was like, I'm really excelling at this, you know, with a physician father, with my parents running the clinic. I was super exposed to medicine very early on. Me and my siblings were always around patients, education. My dad is a teacher. And you know, first I didn't know that I was going to follow this field of medicine. Once I realized this is really what I want to do, I always wanted to become an OBGYN. And I spent 7 years actually being in every delivery room for natural delivery, water birth, with my OBG and my pediatrician. So I was really interested in doing that. And then internship came around, and after 3 months of rotating in OBG, I was like, I really don't think I want to do this for the rest of my life, which was okay. I spent 7 years of really pushing myself through med school on adding more nights and shifts on OBG, but realized that really that was not it. And on my thesis, before I graduated from med school, I joined forces with a neonatologist that was studying the placenta. And I studied so many placentas. We were dyeing the irrigation of the placentas just to see how it actually goes through. This was for twins with one placenta. So little did I know that in the future I would be working with perinatal tissue, and I would be growing cells from perinatal tissue, and placenta being one of the biggest parts of perinatal tissue. So the future is just bright, and the future just gives you different options. Once you think that you're going to do it this way, believe me, life can give you so many turns and curls that you'll have to adapt through them. So this is where I am today. We are a Organocell, the company that I co-founded and I'm the chief science officer of, is a publicly traded biopharmaceutical company committed to the research, development, and manufacture of new biologic medicine. And we really focus on those current and potential regenerative therapeutics. Organocell is located in the CIC at Miami. This is the University of Miami building. And we also just recently opened our in Willits Healthcare Center in Colorado Basalt, our manufacturing facility. So we have 2 facilities now. We're registered with FDA as a human cell and tissue establishment. And I give always all the pros to my team. I am very, very proud to say that we are from all over the world, and we come with all of our beautiful cultures into pushing one agenda, which is really trying to find the next generation of therapeutics, because medicine has offered us up to this point certain solutions, but there are other things that medicine has not found yet, and this is where we are really honing our specialty on. So regenerative medicine. When I went to school, regenerative medicine was not being taught, as a subject. I don't know right now if it really is taught. The world is evolving, and really the future of this field is we are just in the infancy of this field. So it is the branch of medicine specializing in replacing, engineering, regenerating human cells, tissues, or organs. When people think about stem cells, they think either we're going to grow an organ in a petri dish, but there's so many other things with regenerative medicine where we can actually regenerate cells or tissues, not specifically only the organs that we want to implant. And the beauty about this, and what really drives me daily, is how we can optimize the body to heal itself. So before, this was super theoretical, but now we really have so much research to support that ability that the body has to regenerate itself if you are able to deliver a specific message. So in regenerative medicine, to make it a little simple on how to understand this field, we have 2 categories. Either we use an autologous source or an allogeneic source. Autologous meaning auto is your own, so it's the ability of taking cells from your own body and implanting them in your own body. So you are the donor and you're the recipient. In the allogeneic side is using another human as a donor and you're the recipient. So you are using another human as the source. And here are some examples of autologous therapies I'm pretty sure you've heard about, and I'm going to start with the last one, the peripheral blood, where most of you have heard about PRP, or platelet-rich plasma. That is by taking blood from your free-flowing blood and being able to separate it and just collect the growth factors out of the platelets. Other examples of autologous therapies also come from adipose. This is the derived stromal vascular fraction. There are stem cells on those, or progenitor cells in the world of stem cells. One of the gurus is Dr. Kaplan, and even though he coined mesenchymal stem cells or medicinal stem cells, he now calls it progenitor stem cells or progenitor cells. Also, bone marrow carries a lot of hematopoietic stem cells. Those are the ones that derive all of the bone-related, the blood-related cells. And so this is an example of autologous, and it's being used daily in the US. Around the world, we are using autologous therapies. And then we have allogeneic therapies. So after 15 years of working with an autologous therapies, we really focus on looking for better cells or healthier cells that don't come from usually who's seeking for a treatment are patients that are in a lot of pain, that are older, and therefore the results that we used to get were not consistent. So we look for better cells. And this is how the world circled me back to the placenta. And so in allogeneic therapies, in this example, I'm just going to talk about those perinatal derived cells, where all of this tissue is coming from, it's a perinatal tissue that's being donated, right? So the way I always say, it's like the way that you donate blood. You always have to qualify those donors, and those donors that we use and the industry is using, they come from consenting mothers delivering full-term healthy babies by scheduled C-section. So we're talking about, these are tissues that are being procured in an OR after qualifying the mothers and the babies, making sure that everything is healthy, there's no viral infections present in the mother, and they are deemed for donation. This avoids any ethical and moral concerns, because I know that when you say the word stem cells, many people think you're talking about aborted fetuses or unethical ways to collect the tissue. But know that there's, as you can see, so many other ways to collect the stem cells, right? And they've been tested and proven since 1910, imagine. And there's many case series that have been documented. And now we formally are really going under double-blinded placebo-controlled trials under FDA. So after, as I'm saying, 15 years of working with stem cells, we have now developed a drug that doesn't contain stem cells anymore, even though that is our field, even though all of my senior researchers are cell professionals, right? They are in the stem cell world. We have now enter a different phase in regenerative medicine, and hence the title of my talk, because before we did not know how cells communicated with each other, or we didn't know how specific it was. So I'm going to walk you through what it is, the future of what we have found on perinatal derived tissue, specifically this drug that we are carrying our trials with called Zofit, which is manufactured under CGMP compliant manufacturing conditions under an ISO 7 clean rooms. And where no longer do we keep stem cells in our drug. We are maintaining a soluble protein concentration of approximately 1.8 milligrams per ml. And Sofin consists of 300 different cytokines, chemokines, growth factors, and these are signaling molecules that are going to target specific pathways. And I'm here as just a representation of some of the most expressed proteins that are present in Sofin. As I'm saying, maybe I didn't specify it correctly, but Sofin is coming from donated perinatal tissue. So I hope you're following this. So most of these proteins are pro-angiogenic, anti-inflammatory, immune modulatory, tissue repair, and pro-metabolic. This is the protein composition. Besides the protein, there's also a hyaluronic acid component. I don't know how much you know about hyaluronic acid, but it is a glycosamine glycan component of the extracellular matrix, and it's incredible for wound healing. It also has the key to start repairing, and it is a great hydrator. So right now, hyaluronic acid is being used in orthopedic conditions as just injections of hyaluronic acid inside of the joint, just for lubrication. But in the aesthetic world, is being used as direct injections just to skin, just to moisturize the skin a little bit more. But as you can see, it is a very important component of extracellular matrix. So we do quantify hyaluronic acid on our product in every single batch. But the start of the show is this extracellular vesicles, or exosomes, which is how my talk, the title of my talk is how cell waste really is becoming the next frontier of regenerative medicine. So I would say decades ago, we were able to see that the cells would secrete or excrete these lipid bubbles, which we call, and we very known as, extracellular vesicles. But these extracellular vesicles were not being able to be characterized, or you cannot actually see what's in there. So the way that I was taught in medicine, and I don't know how you are taught in medicine right now, but they were considering garbage being excreted out of the cells because we were not able to actually see what's inside of these lipid bubbles. But in the last decade, we have done such an incredible job of understanding that we need to actually see what's inside of them. So exosomes, which are the smaller fraction of these extracellular vesicles, have an endosomal origin. So they are produced inside of the cell, and they're released by every single cell in the body. And they're carrying communication. They are the ones that are having the information from one cell, and that's being sent out not only to neighboring cells, but to every single cell out there in the body with a specific message. And it depends if that cell, what type of cell it is, you can be expressing a message for regeneration or a message of inflammation or a message of a pathological problem. So exosomes are the ones that are working in information for recipient cells. They are secreting that information from one cell to the other. And depending on the origin, they can be transferred and can be altering the fate of the cell, of the cells that are receiving this message. As I mentioned, exosomes are the smaller extracellular vesicles, which are right now deemed in between 40 to 200 nanometers. This is a little more schematic picture of how they are produced inside of the cell, inside of the endosome. It gets the message gets collected as cellular proteins, messenger RNA, microRNA. There's enzymes, there's proteins, and there are surface markers that are unique to each of these lipid bubbles. They go out as a message, and then they are received in the target cells. In any single cell is also absorbing these exosomes or extracellular vesicles with specific information. So the source of your cell will be producing those specific messengers that are carrying an information that will trigger a response on the target cells. And this is what we have heavily invested our time and efforts to understand, because this is what we are pursuing as a drug, as a biologic drug that will be hopefully treating or helping to improve the quality of life of so many conditions that don't have a solution yet in current medicine. So how do we do it? We got to make sure that we quantify every single lot these extracellular vesicles. We are right now in an average of 200 to 400 billion exosomes per ml or nanoparticles per ml. We do not only quantify the amount of them, but we also quantify the size of them. As I mentioned, they have to be between that 40 to 200 nanometers to be called an exosome. And I want to show you what we didn't have before, and we could not quantify these ones and actually see what they were. Now we can count with this technology called the NanoSite NS300, where you can actually see these particles. It's a high resolution of particle size distribution on the number weighted basis on a concentration analysis of a 5 micron particle. So this is how we run every single batch. They are being averaged by the reflection of the light being quantified and also the size of these refractions. But quantifying is not enough. We gotta do more characterization and understanding where they're coming from. Remember, I told you they have surface markers that tells us which cell are they coming from. So that's very important. And another very important thing is to understand what's inside of them. So we've done extreme characterization of them. More of that is on all of our published articles, so I will definitely leave you with the titles of them. But this is what we have accomplished in the last 3 years, going from we were studying for COPD, a lung injury, and COVID happened, and that lung injury that this COVID infection deployed, very similar to what we were looking or studying as a preclinical data on bronchopulmonary dysplasia. So we're able to apply for an IND with FDA. We got the approval, and we are actively enrolling. Then we study in the first year of the pandemic, first to second year of the pandemic, we apply for single INDs, which are emergency compassionate INDs per patient, and we're able to understand different severities of COVID and how our drug can impact in those types of severities. So we did just the ones that recently got infected, the moderate to severe, patients in ECMO, patients in ICU, and then the COVID long haulers or COVID sequelae. There's so many names right now for COVID long haulers. So we have right now 2 sites that are active, that are ready to enroll on our COVID long hauler trial. Very excited about that. Launching already our COPD trial. Very, very, very excited about that because we've been working for 4 years on getting all of that going while got hit with the pandemic, and then decided to, switch things. So we're back in track on making sure that our pipeline gets moving along with COPD. And osteoarthritis of the knee is another one of the disease that we're studying. There's many others that are coming. Our pipeline keeps growing. Our drugs or biologic drugs that we're manufacturing are also growing. So very excited to keep going and keep telling you what we're doing. I did bring a case study, one of our publications from a long hauler, a long COVID patient. This is, as I mentioned before, this is an FDA approval for one single patient. This is a 55-year-old male that has been hospitalized for 7 weeks due to mild to moderate acute respiratory distress syndrome due to COVID-19. During the hospitalization, he was admitted twice to ICU. He was never intubated, and then he was discharged. The patient at discharge was physically limited due to the severity of the conditioning. He had shortness of breath while performing limited activities of daily living, like the smallest things like eating a meal, showering, walking short distance from his bed to the bathroom. He was just short of breath, fatigue. And if you are a long hauler or you have a loved one that is a COVID long hauler, you know how devastating this is, because this patient, 55 years old, pretty young, pretty active, and not being able after discharge to do this simple amount of things, very, very frustrating. Chest x-ray image revealed a bilateral pneumonia at baseline. Baseline for us was when we took over the patient, meaning he was already at home. He was already discharged from the hospital. This is just a example of what we do for this specific trial. For this patient, we had to have a confirmed COVID-19 positive test. Just if we are treating COVID long haulers, FDA does require a COVID positive test. Patient is signing an informed consent. We are taking demographics, medical history, medical exam, chest x-ray, and then laboratory testings. Because our drug is looking for specific targets, which is trying to prove that our drug is anti-inflammatory and immune modulatory, we are focusing on this specific things that we're testing, right? So IL-6, TNF-alpha, D-dimer, CRP are one of those biomarkers that we are really looking in all of our trials to see how much inflammation the patients had when starting our trial and how can that be modified. CBCs and differential complete metabolic panels. And all of this we are doing at day 0, 4, 8, 12, 21, and 28. The days that we give treatment for these patients are day 0, 4, and 8. Sulfin is given as an intravenous infusion on 100 cc's of saline. And we are looking for adverse event assessment. Infusion is associated, all of these things are being captured so we can present. So going forward with after all of these 28 days, on the first day of treatment, the patient's O2 saturation remained low at 96. Besides the saturation itself, it was the fatigue and not being able to do those small things, right? By day 8, as I mentioned, treatment was done 0, 4, and 8. So at the third day of treatment, before the administration of third dose, patient reported no more signs of fatigue and shortness of breath. O2 saturation improved to 97 to 98 after that first and second dose. And after 28 days, x-rays are presented and we see no more pulmonary abnormalities. And the best thing was the patient was able to go back to his work. He was a physician, a surgeon, so he's back at the OR in 1 month. But again, this is one case study. We are studying for a double-blinded, placebo-controlled, randomized trial, multi-center, and this is what we are launching right now. So I just wanted to leave you. This is our publication for you to see a little bit more on what how the design of the trial, what we were looking for. These are other publications that me and my team have produced. And I always end up saying this, and through the pandemic and through those hardship moments that we face when we get diagnosed with things that we never ever expected to be diagnosed, to never give up. Don't ever give up. Science will show up. We are working towards finding solutions to problems that really don't have and have not been addressed in medicine. So I can't wait for you all to join the forces on this field, because it's such a beautiful field, such a incredible field, a field that really needs more and more of us. A little bit more of our company. Please follow us at Organicell.com for information. I am personally at Mari Mitrani in most of social platforms. And I am open for any questions and discussions you might have. Let me see if I can stop this.
Thank you, Dr. Mitrani. It was a great pleasure to hear you talk about regenerative medicine and research. The insights provided by medical research truly shows promise to lessen the impacts of a greater health problems. Your talk really emphasized the importance of research practices. The size of participation during your talk is testimony to how thrilled everyone was to listen to you. Everyone present here learned a great deal through your talk. We will be taking a couple of questions from the audience now.
Thank you, Peter.
So the first question I have here is, are the outcomes of therapies you mentioned the same for different ages, or will it change accordingly?
Yeah, so that is a great question. That is why we switched from an autologous regenerative cell source to a perinatal derived cell source, looking for a more consistent result. In autologous, you are dependent on your donor, which is your patient. And as I mentioned before, those donors or those patients that are coming already with a lot of things, right? But when you're talking about perinatal tissue, that is at birth. So it's day 0. So those cells and that information that those cells are producing, the extracellular vesicles and the message that's inside of those extracellular vesicles are really, really young and new and ready to reconnect everything. So that is why we decided to focus on perinatal tissue, and therefore the consistency of results now that we are seeing are even like that. We can really tell and look for those, as I mentioned, biomarkers, for example. So we did an expanded access trial for COVID active COVID, and those biomarkers are consistently going down besides all of the symptomatology.
Thank you, Doctor. So the second question that we have is, what is the criteria or indication to use Zofin treatment in COVID-19 patients? And were there any limitations to use Zofin drug in COVID-19 cases?
So again, Zofin is a brand new biologic that's being tested. So it's under investigational new drug application. And the way that we designed the trial is specifically for lung, right? So for the lung injury, so for ARDS, acute respiratory distress syndrome due to COVID. And limitations, it was a little hard to navigate the reality that we have in healthcare now, where even though we did have the drug as a new drug, it was tested in many patients. Some facilities did not allow these new drugs to be, even though we can have an FDA approval, some facilities and their own rules and regulations did not allow us to come into their facilities. And we had several friends of ours that had family members that lost their life without being able to given the opportunity of trying it. So the limitations were more on rules and regulations more than the drug itself.
Thank you, Doctor. The next question is, what are the expectations of Zofin on COPD patients based on the initial information that you have?
Yeah, thank you. So our work is all of our preclinical data is conducted in a BPD model, bronchopulmonary dysplasia, where neonatal rats are exposed to oxygen way earlier than, or bronchopulmonary dysplasia is when babies are born earlier than they are supposed to, so their lungs are not mature and they're not ready to that contact of oxygen. So that is what we studied in lung injuries specifically. So very excited of what the future will hold. And as scientists, I can't predict the future more than us having the theory of what we expect to happen with this, not only in vivo with animals, but in vitro, specifically on lung cells, and what we've captured, seen in this models, where there will be better alveolar interaction and hopefully regeneration of the tissue, less inflammatory response on the alveoli itself, and more vascularity around the area. So this is what we're expecting. Soon you'll hear, we're a public company, so soon you'll hear when we're ready to enroll, which is coming along any second now. But moving forward with our site to go there.
Thank you, Doctor. We have very interesting question. For autologous therapies, how safe are these therapies on the long term, taking into consideration that stem cells survive much longer than ordinary cells, increasing the chance that they might accumulate genetic mutations, as well as the chance to become tumors?
I just need you to confirm that you can see me and hear me,
Yes, we can.
because I just have a black screen says that you lost connection. Okay, great to know. So autologous therapies are extremely safe because you are the donor and you're the recipient. One of the biggest questions usually is, are they going to become a tumor? And that we've only seen with very early embryonic stem cells. No other, not perinatal cells, no autologous cells have shown to become or be pro-tumorigenic. And even though you are taking from a older adult that has an issue or a problem, we see that if you do collect the correct amount and go through the protocol of preparation
So it seems that Dr. Mitrani has lost internet connection. Oh, there you are.
See, I had that message and I think it went off. I don't know up to what point you heard me, but yeah, autologous stem cell treatments are extremely safe because they are coming from you, and you're the donor and the recipient. No chances of no teratogenic chances from autologous, and only embryonic, really early embryonic stem cell research has shown that they can be teratogenic. So autologous is pretty safe, even though, as part of the question was, even if that patient is carrying a specific disease, the stem cells that are being so the body has stem cells, a big pool of stem cells, just dormant in the body. So if you have the ability, the correct protocol to collect them, manufacture or clean them from the environment, and then the protocol to inject them or reintroduce them, they're very, very safe.
Thank you, Doctor. To be respectful of everyone's time, we will be discussing one last question. Could you give us a brief insight on how the FDA approval process works for young aspiring researchers to know about it?
Yeah, I mean, that is a great question. And again, coming from a third world country, a small Latin American country, and being faced with the reality that we were able to collect all of our preclinical data, and our research scientists had produced all of this paperwork, it was really nerve-wracking to really go through that first trial. Thank God we have an incredible regulatory team that consults with us, and they've been working with FDA for many years, so they know exactly how to approach what it needs to be done. FDA has been nothing but extremely responsible with our team. In less than 30 days, we got our FDA approval, but it's not because they took it easy. It's because we really had all of the information that was needed. So even though it's kind of scary as first time, or even the third, fourth time, to be presenting to FDA, if you're just following everything that they are asking for, then there's no need to be afraid of that task, right? You all are right now taking test quizzes and going through rounds, right, and presenting patients realities in front of your treating physicians, and it's kind of that that times 100. But what has worked for us is really being proactive on how much information we have beforehand. And FDA has 30 days to respond, and they respond. There's 3 different brackets of FDA that respond to you on. One is the chemical side, one is the clinical side, one is the regulatory side, and they all have questions for you. As soon as you receive those questions, prepare for answers and keep moving on. So there's no clinical hold on those INDs, and then you're ready to have an IND approval. And just to give you with a little bit more note, once you have an IND approved, then you are looking for an IRB approval. You're also looking for sites that know how to conduct research, and you have to engage a CRO that's going to manage your trial. And unfortunately, all of this doesn't come too cheap. It's extremely, extremely expensive to conduct these trials, but and one of the reasons we are a public company, just because we want to really roll this through regenerative medicine, hopefully through our mats really soon, where we expedite the approval. So please keep us keep us on your refresh and look for what we keep doing.
Thank you, Doctor. So IJCR, being a research-centered organization, we always pitch this question to all of our speakers. We have a lot of attendees here that are medical students and are trying to get into research. And it's always a bit overwhelming and confusing getting into research as students in medical school. The question here is, what advice do you have for young researchers?
Yeah. Yesterday a friend of mine said, what is a physician scientist? Aren't all physicians supposed to be scientists? And I was like, well, that sounds really good. Yes, they should all be conducting research, but not everybody at the end ends up conducting research. And it's just a matter of, and for me, I'm always embracing my little kid energy and always asking those questions and how I'm going to answer for those questions. So in any field you are, it really doesn't matter which field you end up doing. We do need to collect more and more data, and just asking the questions, the simple questions, the questions that you'd never got an answer for, those need to be answered. And starting there, always trying to look for mentors, right? Just bugging them and saying, when can I have an hour with you? Can I just shadow you? I will be just on the side. I think those are easy ways to entry, and then you'll figure it out. You don't have to be the best writer, because you can work with other people that are great writers, but you having the idea of what you want to do. And for me, especially being an MD and being thrown at the reality that we are doing research in a field that's so new, gave me the perspective of the clinical side. I wanted to go to clinical as fast as I could. And while all my PhDs are just more focused on, the lab benches and creating all the in vitro, for me, I had the importance of, it's time to hit the public, because we need the solutions really fast. So having your reality, having your culture, being able to speak to your community, all of that gives you a bigger ability to impact research and therefore impact the world.
Thank you, Dr. Mitrani. Your insights were truly valuable. Thank you for your inspiring work and for paving the way for both aspiring and current healthcare professionals. Thank you for your valuable time and effort to make this webinar possible. We have over 350 attendees that have joined us across all live streaming platforms.
Oh, wow.
To everyone in the audience that joined us today, thank you for taking out time so we can be synonymously present here. I hope you leave this webinar full of inspiration and new morale. On behalf of the whole IJCR team, we wish you well and hope to see you next time with yet another exciting webinar. Thank you.
Thank you, Peter. Thank you, everybody.